In this video, we'll be talking about the concept of embryology, and we will be introducing ourselves to some basic facts and problems of science of embryology. So the name embryology is somewhat misleading because it literally means the study of embryo. That is what it means. Now, because embryo denotes the juvenile stage of an animal while it is still contained in eggs within the egg membrane or inside the maternal body. A young animal, once it is hatched from the egg or is being born, ceases to be an embryo and it will escape the sphere pertaining the science of embryology. So if we were to keep strictly to the exact meaning of the word, although birth or hatching from the egg is very important occasion in the life of an animal, it must be admitted that the process going on in the animal body may not profoundly different before and after hatching from an egg, especially in some lower animals. So it will be an artificial to limit the studies to the study of the juvenile forms of an animals. So we can literally say, since we are not studying it only in the juvenile stage, we can say that it is the study of life history of animal as a whole, and accordingly to the interpret the content of science of embryology as the study of development of animals. So I can say that embryology is the study of development of animals. Now, the word development. The word development has to be qualified in turn in the sphere of biology which we are concerned here. What is development? You see, development is used with two different meanings. It is used to denote the process that are involved in the transformation of fertilized egg or some other rudiments derived from the parent organism into a new adult individuals. Also, the term development, however, may be applied legitimately to the gradual historical transformation of life, starting with simple form, which is organic life on planets, and development of the first type may be distinguished as individual developments, or it can be called ontogenic developments. Ontogenic developments. If you understand. But the development in the second type that I mentioned, it can also be called the historical development of a species, that is called the phylogenetic development. So these are the two types of development that we have. Either it is dealing with the historical development, or it is dealing with individual development. You understand? And that is ontogenic development. That is development of individual, like organism individually, or historical development, that is phylogenetic development. So we can therefore interpret embryology as the study of development of animals, and development is used to denote the process that are involved in the transformation of fertilized egg or transformation of some other rudiments derived from the parent organism into a new individual. You understand? And that is what we refer to as either ontogenic development or phylogenetic development. So we can also the development of a new individual occur in phases. Therefore, embryology find its application in stem cell research, medicine, and genetics. There are now phases of embryology. Now, look at this slide that is saying here. The ones I just mentioned are just introduction. So, embryology is the study of developmental event that occurs during the prenatal stage. And this branch of biology and medicine is concerned with the study of embryo as well as their development. So, it is studying two things: the embryo as well as their development. So we have two periods. We have the embryonic period and we have the fetal period. But this is talking about human embryology. But we are not going to human embryology yet. We are still dealing with general embryology, introduction to embryology, where we'll be talking about some animals embryology like amphibians, frogs, amphibians, aves, amongst others. Understand? So we now have different stages of development in embryology. So I call it developmental phase. Let me just write it somewhere around here. We have the first one to be gametogenesis, the second one to be fertilization, the third one to be cleavage, the fourth one to be gastrulation, and the fifth one to be organogenesis. Now, what is gametogenesis? Let us be taking it one by one. Gametogenesis. So let us take this one. Gametogenesis. This is the formation of gametes. Formation of gametes. And these gametes can either be what? Sorry, gametes, not gates. Gametes. And these gametes can either be what? Can either be female gametes, called the what? Called the ovum, or the male gametes, called the what? Called the spermatozoon. So that is gametogenesis, formation of gametes. Now, like I said it involves the formation and ripening of the female reproductive system, female gamete, which is called the ovum, or the male gamete that is called the spermatozoon. So it is a biological process in which a diploid precursor cells, it is a biological process in which a diploid precursor cells is converted, or it undergoes cell division, both meiotic cell division. It undergoes, let me just say, it undergoes cell division, both meiotic cell division and mitotic cell division. You understand? And differentiates to form an haploid gamete called the hen. So this is what we call the diploid, and this is what we call the haploid. If you have checked one of my videos on spermatogenesis, you will understand how a diploid cells is turned into the haploid cells, but I will just try as much as possible to explain that very briefly. So we have a we have a something like this, that is a diploid. So N, it undergo a meiotic cell division to form a what? To form N. You understand? To form N, which in turns undergo division to form haploid. Also, this one undergoes cell division to form haploid. And we're going to be having what? We are going to be having spermatozoon. I'm just using this one to represent it. We are going to be having spermatozoon. You understand? A mature spermatozoon. So this is what we call what? Something like, we call it spermatogenesis. So the formation of spermatozoon is called what? Spermatogenesis. And the second one is, we're going to have a primary oocyte, that is diploid, and it is going to separate into N, which is an haploid, and this one, it is given an haploid cells that we call the what? That we call the ovum, and that is what we refer to as what? Oogenesis. So this is the first step in embryonic development. The first step is what? Spermatogenesis, is gametogenesis. You understand? Now, the second one is now fertilization. So fertilization is the process in which a sperm cell fertilizes the egg cell for zygotes to be developed. You understand? Or for the formation of zygotes. So the sperm cell that is being produced here is going to fertilize the egg cell that is being produced here, and we can say that this egg has been what? It has been fertilized. We can say that this egg has been fertilized. That is basically what fertilization is. Now, let us now go to cleavage. So, cleavage is a process in which a single cell, like a single cell, divides again to give rise to large number of cells. So, the third one is cleavage, which we'll be talking about, in which a single cell divides to give rise to large number of cells, that form a multicellular organisms. And the third one is gastrulation, and this is the process of displacement or invagination of cells to form a what? To form a trilaminar structure called the what? Trilaminar structure called the gastrula. So this is basically organogenesis is the formation of organ, which we will still like try to talk about later in the future. So I've introduced you to the concept of gametogenesis, the concept of cleavage, fertilization, gastrulation, and organogenesis. Now, that is what they are trying to say here. Fertilization is the process of fusion or union of spermatozoon with a mature ovum. You understand? It is also known as conception, fertilization, or impregnation. And it produces a mononucleated cells called the what? Called the zygote. Fertilization produces a mononucleated cells called the zygotes. And after fertilization, we now have embryogenesis. That is the formation of the embryo. And the first one in embryogenesis is the cleavage, which is a series of rapid mitotic division in which cell divide without growing. The cell continue dividing without growing. It still remains the same. You understand? The only thing that happened there is just division. You understand? You will get it much better when we get to cleavage, when you get to the concept of cleavage. The third one is gastrulation. Gastrulation is a phase of early embryonic development in which most animals, a single layer blastula is reorganized into a trilaminar structure, like I have mentioned here. A trilaminar structure, I think I mentioned this somewhere around here. Yes, a trilaminar structure called the what? Called the gastrula. That is what they are trying to like say here. So it is given a what? A trilaminar structure called the gastrula. And these germ layers, they are referred to as the outer ectoderm, the middle mesoderm, and the inner endoderm respectively. So the next one is organogenesis, and that is the production and the development of organ from these three germ layers. That is why I said embryology is very applied in stem cell research and medicine, because you see this ectoderm, it is going to give rise to some structure like the epidermis of the skin and the neural tissue. The endoderm is going to give rise to structure like gastrointestinal tract and respiratory tract. And the mesoderm is going to give rise to structure like urogenital cells, urogenital system, circulatory system, supportive modular system, and skeletal systems. That is, so you see that each of these germ layers, they have what they form, like an organ at which they form, and that is what we refer to as what? Organogenesis. That is the development of organ from the three germ layers that was given initially during gastrulation, and that is what we have here. Now we now have types of eggs that we'll be talking about. We have the isolecithal egg, also known as the homolecithal egg, and these represent egg in which yolk are equally distributed. So let me just write it here: equally distributed. Or let me just write something around it. So we have the homolecithal egg, or homolecithal, sorry, homolecithal egg. In this egg, yolk are equally distributed. And we have the mesolecithal egg, mesolecithal egg, mesolecithal egg, and this one, yolk are moderately like meso, moderately like middle, moderately distributed. Then we have the telolecithal egg, telolecithal egg, and in this case they are found in birds and shark, and those ones that they have enormous amount of yolk, enormous amount of yolk, you understand. And we have centrolecithal egg, we have the central lecithal egg. Those are the ones in which most of the parts of the egg are covered with yolk. You understand? Most of the parts of the egg are covered with yolk. And if you can see it here, we have the telolecithal egg. Can you see? It has n amount of yolk. Central lecithal egg. Can you see? The central part of this thing is covered with yolk. The scoida. We did not mention the scoida in class. So homolecithal or isolecithal egg are those ones that are evenly distributed. So these are the types of egg that we have. Now let us talk about cleavage. So basically, what is cleavage? Cleavage is a series of cell division that occurs without cell growth or gene expression, which occur in early embryogenesis. So we can say that cleavage is a process of progressive subdivision of zygotes by mitotic division into an increasing number of cells of progressively decreasing in size. Now, the consecutive division of cells results in the heap of cells called the morula. So we are going to have something we call the morula. Now, what are they trying to say here? What are they trying to say here is that you see cleavage, it is a series of cell division that happen in rapid succession. You understand? It is a series of cell division that happens in rapid succession which transform a single cell into a multicellular organisms. You understand? So what happened here is that a single cell is going to be transformed into what? A multicellular organism. Multi-cell. Now, how does it happen? That is the question. You see, what is going to happen is that we are going to have something like this. Though we have not get there yet, but let me just quickly say something about it so that you will not be confused. When I said cell continue divide, but there is no growth. You understand? It does not grow. Like there is no increase in the size of the cell, and cell continues to divide. So if you have something like this is a cell, right? Now, this is having 46 chromosomes. You understand? So what is going to happen is that this single cell is going to give rise to this, and it is going to divide into two. You understand? We are going to be having two cells having 46 chromosomes each. You will see that it is a what? It is a mitotic cell division. Mitotic cell division. But if you look at the size of these cells, it does not increase in size. You understand? The size does not increase. Now, which means this is two cell stage. We are still going to have another one in which these two cells, they are going to divide into what? Into four cells. Into four cells. 46, 46, 46, 46. So you will see that there is no growth in the size. There is no increase in the size of the cell. You understand? And that's how they continue to grow. You will now notice that a single cell is being converted to what? Is being converted to a multi-cell. So that is how it continues. We are going to be having something like 8-cell stage. You're going to be having something like 16-cell stage. I'm just trying to draw like, so you are going to have, each of them is going to have 46 chromosomes. You understand? And it is going to give rise to something we call the what? The morula. Although there is a particular aspect of cleavage that we call blastulation. So when it reaches 16 cell stage, we call it the morula. Then it is now going to enter, it is going to develop into something we call the blastula. We are still going to be explaining that later in the future. So what you just have to understand that the basic thing in cleavage is just the conversion of single cell into a multicellular organism. You understand? And this happen by increasing the number of cells, but the size of the cell remains the same. So I will just say increase in the number of cells, but the size remains the same. Or let me say, but there is no growth. because even if you look at it very well, it is decreasing the size of the cell. You understand? It is decreasing the size of the cell, but that is, we are going to be talking about that when we get to. Now, look at this. They said the cleavage stage cells are called the blastome. The total cellular volume of the embryo stays the same. I've mentioned it before, which means there is no growth. The total volume of the embryo, it stays the same, but the number of cells within the embryo increases. And the cleavage rhythm are controlled by the maternal factor present in the cytoplasm of the ovum. And it is from there that we will now be talking about the peculiarity of cell division in cleavage, the chemical changes during cell division in the cytoplasm. That is where you'll be seeing things that are responsible for cleavage in the maternal factor that are present in the cytoplasm of the ovum that is responsible for cleavage. You understand? Now, the cell cycle is biphasic. We know that is the mitotic phase and the synthetic phase. Unlike normal this thing, normal cell division is we are going to be having growth phase 1 or gap phase 1, we are going to be having synthetic phase, we are going to be having growth phase 2, and we are going to be having mitotic phase, and it goes like that. Or it can goes to what? It can goes to G naught, you understand? But in the cleavage, it is biphasic, just two. Instead of four, it is just two. The first one is the mitotic phase, and the second one is the synthetic phase, which is DNA synthesis, which means there is no growth. You understand? There is nothing like the cell is growing in size because the total volume of the embryo stays the same. So you can actually say that the cell cycle of a cleavage is biphasic. You understand? And we can have different this thing. It happens in different animals, like in Drosophila, like this. They say it occurs every 10 minutes, and 50,000 cells is produced in 10 hours, or how many hours? So we have them like that. I'm still going to talk about that. Now, what are now the characteristics of cleavage that you have noticed now? The first one is that this unicellular fertilized cell. The first one is the first one you are going to notice is the single cell or unicellular fertilized cells is converted to what? To a multicellular cells. You understand? To a multicellular embryo. You understand? That's the first characteristic. The second characteristic you are going to notice that there is no growth. You understand? There is no growth during cleavage. You understand? There is no growth during cleavage. And you will also notice that the shape of the cell does not change. The shape of the cell does not change. Although when we get to gastrulation, you will see that the shape of the cell will change. You understand? It become flattens. There will be formation of architrons. They will be pushed inside, blah blah blah, like that. We'll still talk about that. And there is a formation of cavity in the inner structure called the blastocell. There is a formation of cavity called what? Called the blastocell. We are still going to talk about that, called the blastocell. You understand? And the constituent of the cytoplasm is not displaced. The constituent of the cytoplasm is not displaced. They are not given off. You understand? Are not displaced. You understand? So I am looking for somewhere that I can show you how a blastocell looks like. Hopefully, if I can see it. I'm looking for it. Hopefully, if I can see, so that you can see how a blastocell looks like. Or maybe I should just let me just sketch it for you. At the end of the cleavage, there will be pushing of cells from the side to like only one side. Like it will be only one side that you're going to be having cells, something like this. I'm just illustrating, you understand? So we're going to be having some that are lining this particular place. Now you now see this space. That is what we refer to as what? That is what we refer to as the blastocell. You understand? Or the blastocell. You understand? So there will now be formation of the blastula. If you have them like that, that is in cleavage. So I've mentioned the characteristic of cleavage. Let me quickly explain the peculiarity of cell division in cleavage. So cleavage is intricated by division of nucleus. After the division of nucleus, the cytoplasm divide into the two daughter cells without separating, which means there are two basic things that happens in cleavage. That is karyokinesis, that is division of nucleus, and cytokinesis, and that is division of the cytoplasm. But this cytokinesis occurs without the what? Without separation, without separation of the cytoplasm. Without the separation of the cytoplasm. That is, cytoplasm divides, but it does not separate. Now, you see these cells that are formed, like one cells becomes two cells. You see these daughter cells that are formed, we call them the blastomeres. Blastome. So two blastome divide into 4, divide into 8, divide into 16, divide into 32, divide into 64. Now, you see at the early stage of cell division, there is a simultaneous division. You understand? But at the late stage, the synchronization is lost. For example, 2 to 4 to 8 to 16 to 32. After this 32, we are expecting that we're going to be having something like 64 that they are going to do. But at this stage, the synchronization is lost. We are going to be having something like 58, 72. You understand? There will be a loss in synchronization, which means there will not be equal division anymore. Before, there was equal division. You understand? There was equal division. But when it reaches the late stage of this thing, there will not be equal division, which means the synchronization is what? The synchronization is lost. You understand? Towards the end of the what? Towards the end of the cleavage. And you see, cleavage is it is depending on temperature. You understand? It is depending on temperature, which means different animals, cleavage occurs in different animal at different time interval. According to the example we are given, goldfish occurs every 20 minutes, 20 minutes, frog every 1 hour, mammals, it happens every 10 to 12 hours, right? Every 10 to 12 hours. So we can say that temperature affects the time interval of cleavage. So this is where we'll be stopping today. And in the next class, we'll be talking about the chemical change during cleavage, as well as the different pattern of cleavage that we're having. So till the next class, talking about that. Thank you so much.